The spinal cord is the least developed in the neuraxis, following behind the development of the brain. It begins at the spinal medullary junction and runs in the vertebral canal to reach the coccyx. The position soon changes to the adult type by differential development of the two structures. The vertebral canal formed within the vertebral bodies continues the development while spinal cord lags behind. The result is that the spinal cord at birth ends at the level between L1 and 2 of the vertebral canal. The spinal cord then runs down from thence as filium terminale, which is just an extension of pia surrounding the central nervous system and intimately related to the brain and the spinal cord. The filium terminale extends to S2 in the vertebral canal and then becomes invested in dura mater. From this point, its extension to the coccyx is formed by the coccygeal ligament. It has the following morphological features: an expanded cervical portion which is called the cervical enlargement. This is caused by the cervical and brachial plexuses. It also has that lumbar enlargement caused by lumbar and sacral plexuses. Its termination at the level between L1 and L2 of the vertebral canal forms a small protrusion called the conus medullaris. The spinal cord is invested in meninges, which is continuous with the meninges of the brain. They are 3 in number. The pia, which is the most internal and most intimately related to the spinal cord. The arachnoid, which is separated from the pia by a large space called the subarachnoid space. This space is accessible via lumbar puncture. In lumbar puncture, the doctor is interested in either diagnosis or treatment of cerebrospinal disease. Hence, he may wish to obtain cerebrospinal fluid for culture of foreign organisms or obtain the chemistry to determine its glucose or protein content, which may be suggestive of certain diseases. He may also be interested in injecting drugs into the subarachnoid space to treat conditions such as cerebrospinal infections. The commonest of these kind of infections is cerebrospinal meningitis. The subarachnoid space is expanded to the level of S2, where the filium terminale ends. It is more enlarged between the conus medullaris and S2, where the filium terminale ends. Hence, this position is used most extensively to assess the subarachnoid space for lumbar puncture in order to avoid the spinal cord. The dura mater is the most external covering of the brain and the spinal cord. In lumbar puncture, the procedure, which is a clinical one, involves the passing of hypodermic syringe with needle through the back into the skin between L2 and L3, or L3 or 4, or sometimes below L5. But L2 and 3 is used most extensively. The needle passes through the following layers: the skin, fascia, erector spinae tendons, posterior longitudinal ligament, intervertebral disc, anterior longitudinal ligament, ligamentum flavum, dura mater, arachnoid mater, and subarachnoid space. Epidural space. This space exists outside the meninges and specifically outside the dura mater. It is found in an expanded form below S2, where the filium terminale ends and the dura mater merges with S2. Below the level, the space is an expanded epidural space, which can be used for epidural anesthesia for operations on the perineum, and especially during parturition. The cauda equina represents the nerves which issue from the lower parts of the spinal cord, such as the sacral and coccygeal segments. They angulate downwards in order to escape from their corresponding intervertebral foramina. Hence, they form a column of nerves which surround the conus medullaris and accompany the filium terminale to reach the coccygeal ligament and S2 vertebra. These nerves are the ones easily blocked by either a lumbar puncture or an epidural anesthesia. It is equally important to mention that lumbar puncture is also used for anesthesia and is effective for abdominal and perineal procedures. Damage to this column of nerves, that is the cauda equina, may cause what is known as cauda equina syndrome, because they form a column which can be easily displaced by the needle. They do not constitute much danger to the spinal tap, that is another name for lumbar puncture, or anesthesia. The structure of the spinal cord. The spinal cord is made up of the following structures: the covering of meninges, white matter which follows the meninges, gray matter which follows the white matter, and the central canal. The gross structure of a spinal cord is as follows. The spinal cord begins at the spinomedullary junction, placed at the level of a foramen magnum. It then runs from this level to the junction between L1 and 2 vertebrae, where it actually ends as the conus medullaris. It is provided with lumbar and cervical enlargements, and also a slim portion at the thoracic level. Plexuses of spinal nerves are formed at the following levels: cervical plexus from C1 to 4, brachial plexus C5 to T1, lumbar plexus L1 to L4, sacral plexus L5 S1 to S4, and coccygeal plexus S5 and coccygeal 1. A spinal nerve exits from the spinal cord at its corresponding level of intervertebral foramen. It emerges initially as rootlets from two sides: dorsal rootlets, which are more than one, and ventral rootlets, also more than one. The rootlets come together to form the root of a nerve, which is just one, that is T1 or C5, etc. Motor nerves issue from ventral horn of the spinal cord, while sensory nerves issue from the dorsal horn. The sensory nerve appears at the dorsal aspect, while the motor nerve appears at the ventral aspect. Sensory nerves are mainly from the substantia gelatinosa. They form a dorsal root ganglion on the root nerve, at which their pseudo-unipolar neurons have their cell bodies. Note, there is no junction in the dorsal root ganglion except cell bodies of neurons, as opposed to the junctions that occur in autonomic ganglia. This occurs for both somatic and autonomic nerves, so long as they are sensory nerves. Their cell bodies are provided with two processes: a central process and a peripheral process. The central processes enter the spinal cord at the posterior horn, specifically at the substantia gelatinosa for somatic nerves, and the dorsal part of the intermediolateral horn for autonomic nerves. The peripheral processes are connected to interoceptors within the viscera for autonomic fibers, but connected to esteroceptors in the skin and the periphery for somatic nerves. The nervous system is concerned with impulse generation and transfer. It obtains impulses from the environment using sensory apparatus known as receptors. The receptors acquire the impulse and pass them to the central nervous system. We have two morphological divisions of the nervous system as follows: the central nervous system and a peripheral nervous system. The central nervous system has two main organs, brain and spinal cord. The peripheral nervous system consists of all peripheral nerves which may be either motor, sensory or mixed. The nerves associated with the peripheral nervous system can be divided into two types: spinal nerves and cranial nerves. If the nerve is a spinal nerve, it goes to the spinal cord. If the nerve is a cranial nerve, the central process goes to the brain. A ganglion is a collection of cell bodies outside the central nervous system. A nucleus is a collection of cell bodies within the central nervous system. A nucleus is a collection of cell bodies within the nervous system, but a nerve is a collection of nerve fibers outside the central nervous system. A nerve fiber is an axon. A tract or a fasciculus is a collection of nerve fibers in the central nervous system. A funiculus or column is a collection of fasciculus. A funiculus or column is a collection of fasciculi or tracts. There is also a functional division of a nervous system. This is divided also into two types: the somatic nervous system and autonomic nervous system. Somatic nervous system takes care of the voluntary system of the body, while the autonomic nervous system takes care of the involuntary system of the body. Autonomic nervous system is further divided into two types: the parasympathetic system and a sympathetic system. The spinal cord has 31 nerves. They are all called spinal nerves. Motor nerves are provided with multipolar neurons as opposed to the pseudo-unipolar neurons of the sensory nerves. Multipolar neurons have only one axon and several dendritic processes. Their cell bodies lie in the central nervous system. Cervical nerves have 8 nerves from C1 to C8. Thoracic has 12 from T1 to T12. Lumbar has 5 from L1 to L5. Sacral has 5 from S1 to S5. The coccygeal has only 1 nerve. Nerves associated with the brain are called cranial nerves. There are 12 pairs. You have the first nerve, which is the olfactory nerve. The second is the optic nerve. The third is the oculomotor nerve. The fourth is the trochlear nerve. The fifth is the trigeminal nerve. The sixth is the abducens nerve. The seventh is the facial nerve. The eighth is the vestibulocochlear nerve. The ninth is the glossopharyngeal nerve. The 10th is the vagus nerve. The 11th is the accessory nerve. The 12th is the hypoglossal nerve. Cranial nerves are motor, sensory or mixed. They also have autonomic nerves, which are usually parasympathetic. There are 4 parasympathetic ganglia in the head and the neck region. They are as follows: ciliary ganglion, pterygopalatine ganglion, otic ganglion, submandibular ganglion. The brain itself is divided into 2 main parts. There is the brainstem and cerebral together with the cerebellar hemisphere. The brainstem consists of medulla, pons, midbrain, and diencephalon, which is usually referred to as the higher brainstem. Welcome to medulla oblongata. The medulla is the least sophisticated and the lowest part of the brain. It is placed at the lowest level and joins the spinal cord at the spinomedullary junction. It therefore runs from the spinomedullary junction to the pontomedullary junction, where it meets the pons at a sharp edge. It has the following surfaces: dorsal surface, a ventral surface, and a lateral surface. The functions of the medulla. The medulla is a seat of primitive reticular formation which begins to be formed in the lowest vertebrates, such as the fishes. This reticular formation is the most sophisticated motor coordinating unit in these animals, that is, the fishes. In higher vertebrates, however, other centers have taken over the motor control, and in man, the control resides in the highest level of the neuraxis, called the neocortex. The medulla controls the vegetative aspects of the body. Thus, it controls the cardiovascular and respiratory systems and is able to provide a cardiac center, which includes a cardio-acceleratory center and a cardio-inhibitory center. Also, the center for the control of respiratory function is found in the medulla. Thus, if the medulla is dead, there can be no chance that cardiorespiratory function will continue. The medulla provides the largest center for the nucleus of cranial nerves. In the fishes, it provides the center for all cranial nerves, but in man, it only provides the center for the following cranial nerves: hypoglossal, accessory, vagus, glossopharyngeal, and part of the vestibulocochlear nerve, that is, the 8th to the 12th cranial nerves. The medulla provides a highly functioning reticular formation. The medulla provides a center for the nuclei of second-order neurons of the posterior column tracts. These tracts, although primitive, are actually a sophistication over the previous sensory system of the lower vertebrates and the higher invertebrates. Gross anatomy. The medulla is rounded in shape with 3 surfaces as mentioned previously. The ventral surface is marked by the junction of the medulla with the pons. It is provided with a median sulcus called the anteromedian sulcus, which is continuous with the same sulcus in the spinal cord. The sulcus separates the 2 bilateral pyramids which flank it at the upper medulla. Lateral to the pyramids are a sulcus which is continuous with the anterolateral sulcus are the rootlets of the hypoglossal nerve. They occupy this sulcus, which is termed the olivopyramidal sulcus. Lateral to this sulcus is the olive. This upper medullary structure forms a prominence lateral to the sulcus and the pyramid. Lateral to the olive and a bit to the lateral surface of the medulla at its upper part is the inferior cerebellar peduncle. But before this peduncle, we have a sulcus called the postolivary sulcus, which houses the rootlets of the cranial nerves 9th, 10th, and 11th. The 9th is placed superiorly, while the 10th is in the middle. The 11th is placed inferiorly. The remaining cranial nerves associated with the medulla issue from the cerebellopontine angle. This is the junction between the cerebellum, the pons, and the medulla. The nerves are as follows. Vestibulocochlear nerve, which is placed inferiorly. Facial nerve, which is placed superiorly. Nervus intermedius, which is placed in between the two. At the lower level of the medulla, the following are prominent. Pyramidal decussation. These are so clearly evident that it is easy to see the decussation within the anterior median sulcus. The decussation stops at the spinal medullary junction. The olive is only formed at its rostral level. Also, there is no pyramid because the structure that forms the pyramid have begun to decussate at the lower level of the medulla. Dorsal surface. The dorsal surface can be easily described by dividing the medulla into the two parts we had mentioned. An upper medulla, which is also called the open medulla, and a lower medulla, which is also called the closed medulla. The lower medulla has the following important features at the back of that structure. Tuberculum gracilis. This is the swelling which forms the nucleus gracilis within the medullary substance. Tuberculum cuneatus. This also forms the nucleus cuneatus. Accessory cuneate nucleus. The tuberculum gracilis is placed just lateral to the posterior median septum, which is continuous with that of the spinal cord. The tuberculum cuneatus is placed lateral and above the tuberculum gracilis. The accessory cuneate nucleus is placed lateral to the tuberculum cuneatus. All the structures hitherto mentioned can be easily found inferior to the floor of the fourth ventricle. At the upper medulla, the fourth ventricle comes into view. It has the following features. A floor which is diamond-shaped and therefore called rhomboid fossa. A roof which is covered by thylakoid and the inferior medullary velum at the back of the medulla. The rhomboid fossa, and indeed the whole of the fourth ventricle, extends to the back of the pons. Its lower limit or inferior edge forms the calamus scriptorius. At the lateral edge of the calamus scriptorius is the obex. Lateral and slightly superior to the obex is the hypoglossal trigone. Above this trigone is the vagal trigone. At the junction between the pons and the medulla, we have a striae medullaris. At this junction, two lateral recesses of the fourth ventricle are found. They are the foramina of Luschka. Surrounding the junction between the pons and the medulla is the vestibular area, which is found within the tuberculum acousticum. But above the vestibular area and directly behind the pons is the facial colliculus. This colliculus is made up of internal structures as follows. The abducens nucleus, the internal genome of the facial nerve. The lateral surface of medulla has only the inferior cerebellar peduncle. This peduncle begins in the lower medulla and appears on the lateral aspect of the upper medulla to course into the cerebellum. It will carry fibers from the inferior olive to reach the cerebellar nucleus. Internal organization of the medulla. The medulla can be described again from the point of view of two parts, that is, the open, which is the upper medulla, and the closed, which is the lower medulla. We shall take the lower medulla first. The configuration of the lower medulla is similar to that of the spinal cord, but this changes gradually as the spinal medullary junction is reached. Number 1. The formation of the spinal tract and nucleus of the trigeminal nerve at the level of C4 up to the higher cervical levels changes the configuration of the substantia gelatinosa so that it becomes bulbous. This configuration is maintained to higher levels of the medulla. Number 2. The formation of the pyramidal tract and their decussation also changes the configuration of the ventral horn of the gray matter. It is cut off at its bottom by the decussating fibers of the lower medulla, so that The medullary gray matter now looks like a butterfly, together with the bulbous spinal tract of 5, rather than the usual H shape of the spinal gray matter. The formation of the corticospinal tract in the lower medulla leads to the infringement on the gray matter of the ventral horn, so that a reticular pattern is now formed. This reticular formation or pattern is the beginning of a reticular formation. The last is the third point. The fourth point is as follows. The posterior column tract now begins to terminate on their various nuclei, which are the nucleus gracilis, the nucleus cuneatus, and the accessory cuneate nucleus. The second order neurons of the pathway now begin to issue from their nuclei. They will project as internal arcuate fibers, which sweep across the dorsal to the ventral aspect of the lower medulla in order to reach the contralateral medial lemniscus. For these fibers decussate ventrally and form the medial lemniscus of the opposite side. Point number 5. The formation of the medial lemniscus is a feature of all levels of medulla, beginning from the lower medulla upwards. Point number 6. Spinal lemniscus is a continuation of the lateral funiculus, which begins from the spinal cord levels. They include the spinothalamic tract and the spinocerebellar tract. And finally, the central canal is maintained just like it is in the spinal cord. Welcome to the second part of the medulla. Now we shall now talk about the upper medulla. At the upper medulla, the following structures are seen internally: the inferior olive, pyramid, inferior cerebellar peduncle, fourth ventricle, and the cranial nuclei at its floor, and then medullary reticular formation. We shall take these structures one by one. The inferior olive. The olive, which is seen externally at the open or upper medulla, is called internally the inferior olive or the inferior olivary nuclear complex. It has the following nuclear groups: the principal inferior olivary nucleus, accessory dorsal olivary nucleus, accessory medial olivary nucleus. The olive projects 2/3 of the fibers which enter the inferior cerebellar peduncle in order to reach the cerebellum. Hence, its main function is to provide a center for the interconnection between the medulla and the cerebellum. Being inferior in the neuraxis, it is therefore quite primitive. The olive has efferent projections from various parts of the brain and spinal cord. They include spino-olivary tract, cortico-olivary tract from the cortex, and then fibers from the midbrain at the periaqueductal gray and the red nucleus. The fibers coming from the red nucleus are called the rubro-olivary tract. White matter of the olive is called ammiculum olivi. The pyramids. The pyramid contains corticospinal tracts, which are 3 in number. Lateral corticospinal tract, which begins decussation at the lower medulla and ends its decussation at the spino-medullary junction. Number 2, you have the anterolateral corticospinal tract. And the last one, you have the anterior corticospinal tract. The last 2 are uncrossed tracts. Inferior cerebellar peduncle. This begins at the upper medulla and crosses lateral to the fourth ventricle and anterior to it. It is also lateral and dorsal to the olive. It enters the cerebellum and sweeps across the vestibular and cochlear nuclei at the pontomedullary junction. The medullary reticular formation. The medullary reticular formation is the highest motor coordinating center in lower vertebrates like fishes. It projects fibers to the spinal cord as reticulospinal tract, and fibers reaching from various parts of the brain keep it under control. Hence, we have the cortico-reticular fibers. Also, fibers project from medullary reticular formation to the higher cortical centers. These fibers represent the so-called ascending reticular activating system, or ARAS, or reticular formation for short. This formation is responsible for arousal and hence keeps the individual at his very conscious self. It is acted upon by various fibers coming from the neural axis. It is blocked by certain anesthetic agents such as ketamine. The medullary reticular formation is divided into two parts. We have a lateral group and then a medial group. The medial group is further divided into nucleus reticularis gigantocellular and a nucleus reticularis parvocellularis. The last carries very large cells, while the next to it, that is the parvocellularis, has only small cells. The inferior cerebellar peduncle is one of the three peduncles that interconnect the cerebellum with the brainstem. It carries fibers of the posterior spinal cerebellar tract, which form 1/3 of its fibers. It also carries fibers from the cuneo-cerebellar tract, beginning from the accessory cuneate nucleus. But 2/3 of its fibers are projections from the inferior olive in the tract called the olivocerebellar tract. Cranial nerve nuclei are found in abundance in the fourth ventricle at its floor. The hypoglossal nucleus lies in the hypoglossal trigone at the extreme inferior edge of the floor of the fourth ventricle. It lies above the obex and lateral to the area postrema, but below the vagal trigone. It contains nucleus of this nerve which carries general somatic efferent fibers to intrinsic and extrinsic muscles of the tongue, excepting the palatoglossus. It is a new addition to the cranial nerves of vertebrates, of fishes and lower vertebrates like the hypoglossal nerve. It may also carry general somatic efferent fibers to the meninges of the posterior cranial fossa, but this is doubtful. When this nerve is damaged, it will cause an ipsilateral hemiparesis of the tongue. In the condition known as inferior alternating hemiplegia, there is a contralateral hemiplegia and an ipsilateral hemiparesis of a tongue, presumably due to the closeness of the pyramids to the hypoglossal nucleus, so that damage to one may also affect the other. Because the fibers of the hypoglossal nerve are not crossed, we then obtain an ipsilateral hemiparesis of the tongue. The accessory nerve is the 11th cranial nerve. It has two components, a spinal accessory and a cranial accessory. The spinal accessory begins at the level of C2, and its fibers are from the ventral horn of these cervical levels. The fibers enter the cranial cavity via the foramen magnum and joins the counterpart, the cranial accessory, as it emerges from the nucleus ambiguus, which is associated with the vagal trigone. The two meet at the cranial cavity and then separate at the jugular foramen. The cranial accessory joins the vagus and is distributed to the recurrent laryngeal nerve to supply the intrinsic muscles of the larynx. The spinal accessory moves inferiorly to enter the posterior triangle of the neck and disappears behind the trapezius and supplies that muscle and the sternocleidomastoid. The vagus nerve is the 10th cranial nerve. It is very extensive and it is provided for by a special trigone in the floor of the fourth ventricle. This trigone is called the vagal trigone, and it is placed lateral and superior to the hypoglossal trigone. It contains mainly the dorsal nucleus of the vagus, which is the main nucleus associated with the vagus nerve only. Other nuclear groups associated with the vagus nerve are as follows. Number 1, the nucleus of the tractus solitarius. Number 2, the inferior salivatory nucleus. Number 3, nucleus ambiguus. Number 4, nucleus of the spinal tract of trigeminal nerve. The dorsal nucleus of the vagus. This nucleus is only associated with the vagus nerve. It is the specialized nerve for muscle function, smooth muscle function, and therefore it supplies all the areas of the vagus distribution with muscle fibers for the smooth muscles of the airways and the footways. The vagus extends its supply to the gastrointestinal tract after the laryngeal and pharyngeal supply to reach the level of the splenic flexure. Parasympathetic fibers for supply below or inferior to the splenic flexure is via the sacral outflow and not from the vagus. Hence, the vagus is the most extensive in supply and distribution of our cranial nerves. It also supplies the heart muscles with cardioinhibitory vagal fibers, and hence, when damaged, may cause problems with the heart. It supplies all the smooth muscles of the larynx and trachea, together with the gastrointestinal tract up to the splenic flexure. It is placed most medial in the area associated with the vagus at the floor of the fourth ventricle. The nucleus of the tractus solitarius. This nucleus is placed lateral to the dorsal nucleus of vagus. It contains cell bodies of the second order neurons of a taste pathway. They will carry special visceral efferent fibers to reach the gustative nucleus of the cortex at temporal lobe area 43. They have their cell bodies of the first order neurons placed in the ganglion nodosum, which is the inferior vagal ganglion. This ganglion is associated with general somatic efferent and special visceral efferent fibers, while the superior vagal ganglion is associated with the general somatic efferent fibers. Nucleus ambiguus. This is a special medullary nucleus which is associated with only structures derived from the pharyngeal arches. Hence, it carries special visceral efferent fibers. It is placed between the nucleus of the tractus solitarius and the dorsal nucleus of vagus around the vagal trigone. Supplies all the muscles of the larynx through superior laryngeal nerve and also the recurrent laryngeal nerve. The superior laryngeal nerve supplies the cricothyroid and the cricopharyngeus, while the recurrent laryngeal nerve, which was initially carried in the cranial accessory but is later distributed by the vagus, supplies all the intrinsic muscles of the larynx excepting the above two. The nucleus of the spinal tract of 5. This nucleus is the most lateral of all the nuclei associated with the vagus. It carries general sensation of somatic system from the area of the auricle and the external auditory meatus. These are very small fibers whose cell bodies are placed in the superior vagal ganglion. The central processes then run to the medulla and synapse with the second order neurons at the nucleus of the spinal tract of 5. The tract of the second order neurons form the trigeminothalamic tract. They will project into the thalamus and reach the ventral posteromedian nucleus of the thalamus, VPM, where third order neurons issue to reach the sensory cortex. Inferior salivatory nucleus. This nucleus is the caudal extension of the pontine superior salivatory nucleus. As its name implies, it is secretomotor and therefore it carries general visceral efferent fibers to the glands in the foodways and airways supplied by the vagus and also to the glands of the soft palate and epiglottis. It is also the area that special visceral efferent fibers associated with the vagus supplies, for they have scattered taste buds on their surfaces, that is, the soft palate and the epiglottis. The functional components of the vagus are therefore the following. They are 5 in number. GSA fibers associated with auricular sensation, GVA fibers for general visual sensation in areas supplied by the vagus nerve. The cell bodies lie in the inferior vagal ganglion called the ganglion nodosum. The special visual efferent fibers, these are taste fibers from the soft palate and the epiglottis. Special visual efferent fibers for the muscles of larynx derived from the pharyngeal arches. Then you finally have the 5th one, which is the general visual efferent secretomotor fibers to all areas supplied by the vagus, which includes the intestinal, gastric glands, and other glands in the digestive system to provide digestive and other juices. This is the third and the concluding part on the medulla. We shall now start with the glossopharyngeal nerve. This nerve does not have a special trigone in the floor of the fourth ventricle, but its nuclei are associated with the vagal trigone as follows. Nucleus ambiguus, which provides SVE fibers to the muscle derived from the third pharyngeal arch and which is supplied by the glossopharyngeal. That nerve, of course, is the stylopharyngeus. The inferior salivary nucleus. The fibers associated with the ninth cranial nerve supply the glands of the posterior one-third of the tongue and the oropharynx, which are the main areas of supply by the glossopharyngeal. The nucleus of the tractus solitarius carries SVA taste fibers to the areas supplied by the ninth cranial nerve, which are the oropharynx and posterior one-third of the tongue. The nucleus of a spinal tract of trigeminal nerve, this carries GSA fibers which run in the Jacobson's nerve to supply the bony aspect of the auditory tube, inner aspect of the tympanic membrane, and the tympanic cavity. The functional components found in the glossopharyngeal are as follows. There are 5 in number, just like the vagus. Number 1, GSA fibers which run in the Jacobson's nerve. They will supply the inner aspects of the tympanic membrane and the tympanic cavity. Number 2, GVA fibers for general sensation in the oropharynx and the posterior one-third of the tongue. The GVE fibers, they are secretomotor fibers for the glands in the oropharynx and the posterior one-third of the tongue. SVE fibers to the only muscle that is supplied by the glossopharyngeal nerve arising from the pharyngeal arch, specifically the third pharyngeal arch, which is the stylopharyngeus. And the 5th, SVA fibers. These are taste fibers to the posterior one-third of the tongue and the oropharynx. The 9th cranial nerve escapes from the medulla at the upper part of the post-olivary sulcus, above the rootlets of escape of the vagus, which is in turn released above the rootlets of escape of the accessory nerve. The nerve then enters the jugular foramen, accompanied by the 10th and 11th cranial nerves. Together they transverse this foramen in order to escape into the neck region. At the neck region, the 9th nerve seeks for its muscle of supply. After giving off branches to the oropharynx and the posterior one-third of the tongue, it enters the stylopharyngeus in order to supply that muscle. The vestibulocochlear nerve. This nerve is the 8th cranial nerve, and it is essentially 2 nerves, the cochlear nerve and the vestibular nerve. The nerve is the only cranial nerve, or indeed only nerve at all, that is SSA, that is, it carries functional components special, somatic, afferent. It is therefore sensory to the inner air apparatus, such as the semicircular canals, the vestibule, and the cochlear nerve. This nerve has cell bodies of its neurons in the spiral ganglia arranged along the cochlear apparatus of the osseous labyrinth. Spiral laminae. The peripheral processes receive impulses from the hair cells which line the organ of Corti. The central processes of the neurons then issue from the spiral ganglia. They are actually bipolar neurons rather than pseudo-unipolar. The cell processes gather this together to form the cochlear nerve, which approaches the internal acoustic meatus, accompanied by the facial nerve. The nervus intermedius also accompanies it, together with the labyrinthine artery. Its fibers sweep across the superior edge of the inferior cerebellar peduncle at rostral level of the medulla. The fibers then terminate on the dorsal and cochlear nuclei of the floor of the fourth ventricle at the so-called tuberculum acousticum found in the vestibular area. Second order neurons now issue from these nuclear groups to project upwards, sweeping across the medial lemniscus and also the superior olive to reach the trapezoid body, all of which are found in the pontine tegmentum. The fibers pass through the lateral lemniscus to reach the midbrain and terminate at the inferior colliculus of the midbrain septum. Third order neurons now issue from this superior colliculus to project upwards. Their destination will be the medial geniculate body of a metathalamus. From the medial geniculate body, the geniculotemporal tract arrives and terminate on the temporal or auditory cortex. This is at the transverse temporal gyrus of the temporal lobe. It is possible for the fibers to reach the medial geniculate body directly from the dorsal or ventral cochlear nuclei of the medulla of 4. The fibers to run from the inferior colliculus to terminate in the temporal cortex directly. Whatever be the case, the fibers which reach the superior colliculus or the medial geniculate body are responsible for auditory reflexes. Those which reach the cortex are responsible for voluntary mechanisms for which man is well known. All the pathway of the auditory system have several nuclei which occur. For example, you have the following nuclear groups: the nucleus of the superior olive, the nucleus of the trapezoid body, the nucleus of the lateral lemniscus. We also have the brachium of the inferior colliculus, which is the structure that projects fibers from the inferior colliculus to the medial geniculate body. Vestibular nerve. This nerve has its cell bodies placed in the vestibular ganglia. It is responsible for kinetic and static equilibrium. Its peripheral processes reach the receptors, which are hair cells placed in the vestibular apparatus, maculae of truncule and sacral, and also the semicircular ducts, which are the ampullae in the inner ear. The central processes issue from these bipolar neurons of the vestibular ganglia and move centrally, accompanied by the cochlear nerve and the facial nerve into the internal acoustic meatus. It then enters the lower part of the pons and the upper part of the medulla, also sweeping across the inferior cerebellar peduncle to terminate in 4 characteristic nuclei as follows: medial vestibular nucleus, lateral vestibular nucleus, inferior vestibular nucleus, and superior vestibular nucleus. These nuclei have afferent or efferent fibers from all parts of the brain, including the cerebral cortex. Fibers also reach from the spinal cord and the higher cortical centers via the medial longitudinal fasciculus. Efferent projections now issue from these nuclei and project into the spinal cord via the vestibulo-spinal tract and other parts of the neuraxis. Some will project into the inferior cerebellar peduncle to reach the cerebellum. Welcome to the lecture on the pons. The pons is a special relay center for the cortico-pontocerebellar tract. It is responsible for highly coordinated repetitive movements that require skill, extensive control, and intensive training. The pons is placed between the medulla inferiorly and the midbrain superiorly. It is divided into a ventral basis pontis and a dorsal pontine tegmentum. Grossly, it has 2 massive structures on the sides of the ventrally placed basis pontis. The groove or sulcus between the 2 lateral structures contain the basilar artery, which provide pontine branches that reach the pons circumferentially. On the lateral aspect of the pons, we have 2 massive middle cerebellar peduncles, which course forwards and downwards to reach the cerebellum. They are carrying fibers which are second-order neurons of the cortico-pontocerebellar pathway to the deep cerebellar nuclei. Posteriorly, the pons is provided with the floor of the 4th ventricle. The roof of the ventricle is made up of the superior medullary velum, and the fastigium of the cerebellum. Behind the pons, the fourth ventricle has the following prominent structures: tuberculum acousticum, which is part of the vestibular area; striae medullaris; median eminence, which is continuous with that of the medulla; and the fascia colliculus. Internal structure of the pons. The basis pontis. This is the ventrally placed structure of the pons, containing massive corticopontine fibers which terminate on their second-order neurons in the basis pontis. The fibers arise from all the lobes of the cortex. Hence we have the following fibers: temporopontine fibers, parietopontine fibers, occipitopontine fibers, and frontopontine fibers. Frontopontine fibers occupy the anterior position in the pyramidal tract. The fibers terminate at different levels in the pons and synapse on the second-order neurons, which then run transversely to reach the middle cerebellar peduncle of the opposite side. These fibers are termed the transversely running pontine fibers. They will project through the middle cerebellar peduncle to enter the deep cerebellar nuclei, which are as follows: globus, emboliform, fastigious, fastigial, and dentate nuclei. Pontine tegmentum. This carries two important structures. Number one, the pontine reticular formation. Number two, the cranial nerve nuclei associated with the pons, which are as follows: the fifth, the sixth, and the seventh cranial nerves. The pontine reticular formation. This is an extension of the medullary reticular formation, and it consists of two parts: a central tegmental part and a lateral part, which contains the following nuclear groups: nucleus reticularis, pontis, pars, australis. This is the most cranial of the groups. Nucleus reticularis, pontis, pars, caudalis. This is the continuous with the reticular formation of the medulla. And the last is the nucleus reticularis, pontis, pars, interpolaris. This is placed between the pars australis and the pars caudalis. Pontine cranial nerve nuclei. We have facial nerve. This nerve has its nucleus buried in the fascia colliculus, a little ventral to the nucleus of the abducens nerve. It then forms a genu which sweeps across the dorsal surface of the abducens nucleus in order to reach the lateral aspect of the pontine tegmentum, where it runs a ventral course to escape at the cerebellopontine angle, medial and inferior to the point of escape of the abducens nerve. It immediately at its extrapulbar course forms the external genu of a facial nerve before it expands to form the geniculate ganglion. This nervous intermediate part then gives off two branches, the chorda tympani and the greater pretorsal nerve. The chorda tympani enters the lingual nerve and it carries secretomotor fibers which reach the submandibular ganglion and are distributed to oral glands and the submandibular and sublingual salivary glands. The greater pretorsal nerve passes forwards to enter the pterygopalatine ganglion, where it relays, and its postganglionic fibers then enter the nasal and palatine glands. The facial nerve itself proceeds through the stylomastoid canal and emerges at the stylomastoid foramen to enter the face and supply the muscles of facial expression. The nucleus associated with the facial nerve is the motor nucleus of the facial nerve. Other nuclei are the superior salivary nucleus, the nucleus of the tractus sclerotarius. Hence, the facial nerve, together with its nervous intermediate component, has the following functional components, which are four in number. 1. GSA fibers, which supply the auricle. 2. GVA fibers, which are secretomotor from the superior salivary nucleus. They will supply the submandibular and sublingual glands, including nasal and palatal glands, and the lacrimal apparatus via the nerve which hitchhikes in the zygomatico-temporal nerve. Number 3, you have SVE fibers which supply the muscles of facial expression, including platysma, buccinator, posterior belly of the digastric, and the stylohyoid. They are from the motor or nucleus, the motor nucleus of a facial nerve. Then you have SVA fibers for taste in the anterior two-thirds of the tongue via the chorda tympani. The abducent nerve. This nerve has its nucleus placed within the facial colliculus. It is responsible for the formation of the colliculus at the floor of the fourth ventricle. The fibers of the internal genu of the facial nerve sweeps across its normal dorsal aspect and later lateral to it. It escapes, that is the abducent nerve, escapes from the brainstem above the pyramid at the pontomedullary junction. The abducent nerve is surrounded by the paraabducent nucleus. This nucleus is thought to contain fibers which are part of the extrapyramidal system that control lateral gaze movement. Abducent nerve supplies the lateral rectus. Damage to this nerve will cause paralysis of the lateral rectus. Damage to the paraabducent nucleus will lead to paralysis of lateral gaze. The rostral part of the pons begins to change its configuration so that to that of the isthmus rhodencephaly. Hence, certain nuclear groups belonging to the isthmus begin to appear and show themselves in the upper part of the pons. They include certain raphe nuclei and the locus coeruleus. Welcome to the second part of the pons. We shall begin by talking about the trigeminal nerve. The trigeminal nerve has the following nuclear groups in the pontine tegmentum. The sensory nuclear group, which carry general somatic afferent fibers, GSA. They are 1. Mesencephalic nucleus of 5. 2. Principal sensory nucleus of 5, that is the pontine nucleus. Number 3. Spinal nucleus of 5. This is an extension from the upper cervical level through the medulla. Number 4. Motor nucleus of 5, which carry SVE, special visceral efferent fibers to structures derived from the first pharyngeal arch, that is the muscles of mastication and others. The sensory nuclear group is quite massive. It extends from cervical level C4 through the medulla to the mesencephalon. It may therefore be divided into two. A superior group, this is formed in the pons and in the midbrain. It is the principal sensory nucleus and the mesencephalic nucleus of 5. The pontine nucleus or principal sensory nucleus is placed lateral to the motor nucleus in the pontine tegmentum. They both lie in the dorsal aspect of the tegmentum, close to the floor of the fourth ventricle. The mesencephalic nucleus is slightly displaced more dorsally, medial to the superior cerebellar peduncle, and extending upwards into the midbrain. The mesencephalic nucleus contains first-order neurons for proprioception of the face region, supplied by the fibers of 5. The principal sensory nucleus is responsible for fine sensation of the face region, while the spinal nucleus is responsible for coarse sensation in the same face region. The sensory nucleus neurons are pseudo-unipolar, with the cell bodies in the trigeminal ganglion placed at the trigeminal cave of the middle cranial fossa. Their peripheral processes pass through the corresponding nerve divisions as follows: the ophthalmic, maxillary, or mandibular divisions. The sensory nuclear group is arranged in a laminar fashion. Ophthalmic fibers are ventral, maxillary are interminate, and medulla or mandibular are dorsal. Motor nucleus of 5 passes through the portio minor, being smaller than the sensory division which passes through portio major. The fibers remain separate from the sensory division, but join the division distally by entering the medulla, the mandibular nerve, which carries them through the foramen ovale into the infratemporal fossa. The motor division of 5 would supply the muscles of mastication, medial pterygoid, lateral pterygoid, masseter and temporalis. These are the muscles of mastication, and then other muscles derived from the first pharyngeal arch, which are as follows: malohyoid, tensor tympani, tensor villi palatini, anterior belly of digastric, all with special visceral efferent fibers, SVE. The isthmus rhubencephalii. The junction between the pons and the midbrain contains the isthmus rhubencephalii. This means the isthmus of the hindbrain. It is so called because it has a small cavity which joins the cerebral aqueduct superiorly in the midbrain. The isthmus has certain characteristics. For example, the raphe nucleus and lucus coeruleus are found in its tegmentum. Its ventral portion is similar in configuration to the upper part of the pons. This contains prominent transversely running fibers which are equally maintained at rostral levels in the lower part of the midbrain. It has a diagnostic feature, which is the presence of the decussation of a trochlear nerve which takes place dorsally and dorsal to the superior edge of the fourth ventricle at the superior medullary velum, which forms the roof of the fourth ventricle at the back of the pons. This is usually used to determine the presence of the isthmus in cross sections of the brainstem. The superior cerebellar peduncle also begins its decussation at this level. This decussation will continue to lower levels of the midbrain. The lateral lemniscus is now placed lateral to the medial lemniscus, beginning at rostral levels of the pons. The superior olive is placed between them. Gradually, the lateral lemniscus begins to escape dorsally as it enters the tectum, initially into the inferior colliculus before it reaches the superior colliculus. The midbrain is the least developed of all the parts of the brainstem in man. In reptiles, it is the most developed, and it is called the tectum opticum in those group of animals. The gross structure is as follows. It is provided anteriorly with the crura cerebri on both sides. Between the crura is the interpeduncular fossa, which forms a cistern called also the interpeduncular cistern. Laterally, we have the trochlear nerves hugging the sides of the midbrain as they arise from the superior medullary velum to escape from the brainstem and enter the cavernosinus. Posteriorly, the midbrain extends from a junction with the isthmus rhubencephalii to the lower border of the posterior commissure. Anteriorly, this extension is from the isthmus rhubencephalii to the inferior edge of the mammillary body. Internal organization of the brain, of the midbrain. The midbrain can be divided into the following parts: a crus cerebri or basis pedunculi, a midbrain tegmentum, and finally a midbrain tectum. The crus cerebri. This is the extension of the pyramidal tract which runs from the cortex to the spinal cord. In the midbrain, it carries massive corticopontine fibers which are arranged as follows. The cerebral peduncle is one half of the midbrain, which includes the tegmentum and the crus cerebri of that side. The most medial side of the crus cerebri is the medial pes lemniscus. This is followed by the frontopontine fibers, and then followed by corticospinal and corticobulbar tracts, and these are usually centrally placed. Lateral to the corticospinal tract is the parieto-pontine tract, followed by occipito-pontine and then temporopontine tracts. The last part of the crus cerebri at its inferior edge is the lateral pes lemniscus. The midbrain tegmentum. This consists of the midbrain periaqueductal gray matter containing a reticular formation of the midbrain and the midbrain cranial nerve nuclei, which are found at the cerebral aqueductal floor. It also includes other peculiar nuclear groups such as the red nucleus, substantia nigra, and nuclear groups of the reticular formation. The midbrain reticular formation includes the following: nucleus subcuneiformis, nucleus cuneiformis, ventral tegmental nucleus, dorsal tegmental nucleus, nucleus tegmenti pedunculo-pontinus, and finally the nucleus of dark Schwitz. The tectum of the midbrain. The tectum of the midbrain is placed dorsal to the cerebral aqueduct. It contains the inferior and superior colliculi. The inferior colliculus is the relay reflex center for auditory activities. It has a central core surrounded by a cortex. The cortex is further surrounded by the pericollicular gray matter. The superior colliculus, on the other hand, has many zonulations. Its most external part is formed by the stratum cinereum, followed by the next, which is the stratum zonale, and this is followed by another zone which is called the opticum, that is stratum opticum, and the last, most internal zone is the stratum lemnisci. Superior colliculus is responsible for visual reflexes. It projects its fibers via the brachium of superior colliculus to the lateral geniculate body, which also deals with visual reflexes at the level of the thalamus. Levels of the midbrain. The midbrain is usually described in two levels. 1. The level of the inferior colliculus. 2. The level of the superior colliculus. The nucleus of the oculomotor nerve is placed at the level of the superior colliculus, while the nucleus of the trochlear nerve is placed at the level of the inferior colliculus. Hence, the colliculi are used to recognize these levels. The red nucleus. This nucleus is found in the midbrain tegmentum. It is red because it is highly vascularized. It is divided into two parts. Number 1, a parvocellular part, which is called neoruba. The second, magnocellular part, which is called paleoruba. It has afferent and efferent projections to various levels of a neuraxis. Afferent projections of a red nucleus. They are as follows: cortico-rubral tract. This is coming from the cortex to the red nucleus. Spino-rubral from the spinal cord. This is doubtful. And cerebello-rubral tract from the cerebellum via the superior cerebellar peduncle. Efferent projections of the red nucleus include rubrothalamic tract, which is the second order neuron of the pathway known as the cerebello-rubrothalamic tract. This tract begins as the superior cerebellar peduncle, that is most of that peduncle. Its cell bodies are placed in the deep cerebellar nuclei of the cerebellum at the fastigial globus and boliform dentate nuclei. The fibers transverse the red nucleus and then synapse onto their second order neurons, which then proceed to the thalamus, specifically to the ventrolateral nucleus of the thalamus, VL nucleus. Rubrocerebellar tract. These run into the cerebellum as reciprocal connection to the cerebello-rubral tract. Rubobulbar tract runs into the brainstem, that is into the medulla. Rubro-olivary tract enters the inferior olive. Damage to the red nucleus causes what is known as the syndrome of Benedict, which has the following clinical features: tremor, ataxia, coliform activity, to include damage to the ipsilateral oculomotor nerve. Substantia nigra. This is a very important tract in the extrapyramidal system. The striatal tract interconnects the substantia nigra with the corpus striatum. It carries synthesized dopamine to the corpus striatum. When this tract is damaged or diseased, serious impairment of function occurs, leading to what is called Parkinson's disease or Parkinsonism. In this condition, there is depletion of dopamine in the corpus striatum. It may be treated with anti-Parkinsonian drugs or almost permanently cured using fetal tissue transplant. There is a reciprocal tract which complements the nigrostriatal tract, by which the basal ganglia keeps the substantia nigra in check. This is called the striato-nigra tract or strio-nigra tract. Important tracts in the midbrain include fasciculus retroflexus, which is also called the habenulo-interpeduncular tract. This tract is coming from the habenular nucleus and running in the interpedunculae fossa medial to the oculomotor nucleus and sometimes even transversinate. The pretectal area. This area is placed above the level of the superior colliculus. It contains the following. Number 1, nucleus of the pretectal area. Number 2, nucleus sublentiformis. Number 3, interstitial nucleus of Kajal. Some of these are extensions of the nuclei of the midbrain reticular formation. It also includes certain nuclei of the posterior commissure. The posterior commissure is part of a diencephalon, strictly speaking. Oculomotor nuclear complexes. This consists of the following nuclear groups. Number 1, lateral somatic cell, that is nuclear column. It supplies the whole of the extraocular muscles which derive their nerve supply from the oculomotor nerve. Number 2, caudal central nucleus. It supplies the levator palpebrae superioris. The rostral extension of the somatic nucleus provides the Edinger-Westphal nucleus, which is called the visceral nucleus, and which gives off fibers that run into the ciliary nerves and synapse at the ciliary ganglion in the orbit. They supply the sphincter pupillae. Accessory oculomotor nuclei include number 1, interstitial nucleus of Kajal. Number 2, nucleus of Dark-Schwitz. Number 3, nucleus of posterior commissure. The trochlear nerve. This nerve has its nucleus placed in the floor of the cerebral aqueduct and at the level of the inferior colliculus. It then runs to the superior medullary velum where it decussates. It emerges from the brain stem at the dorsal aspect and is the only cranial nerve that does so. It then runs on the lateral aspect of the midbrain to reach the ventral aspect where it enters the cavernous sinus in the cranial cavity. Welcome to this talk on diencephalon. The diencephalon is the caudal part of the forebrain, which is also called the prosencephalon. It is a complex structure with equally complex function and development. It is responsible for the vegetative functions in the human body. Hence, it is the head of the endocrine system, the diffuse neuroendocrine system, and the limbic system. It is also considered to be the largest integrating center for subcortical sensory responses via the thalamus. It is therefore a very important structure in the human brain. Above the diencephalon, no other area is important in keeping the system under check. The diencephalon controls the whole of the autonomic nervous system and has a major function in somatic system, especially as it relates to the sensory apparatus. The diencephalon develops around the third ventricle, and hence its cavity is the third ventricle. Also, its medial aspect is formed by the basal ganglia, and so the structures are interwoven in a complex manner that can easily confuse the inexperienced. Only careful coronal sections, which is presented in this lesson as stained specimen or presented elsewhere as unstained gross brain, can unravel this complex relationship. But diencephalic structures tend to occupy the posterior aspect of the complex of the diencephalon and the basal ganglia, forming the lateral wall of the third ventricle in this position. The diencephalon is divided into the following parts. Number 1, thalamus. This is the most massive and the most highly developed of the diencephalic structures. Number 2, hypothalamus. This is placed ventral and below the thalamus. Epithalamus. This is placed dorsal to the third ventricle and thalamus. Number 4, the subthalamus. This is placed below the thalamus but a little dorsal to the hypothalamus. The thalamus is the head of a sensory system below the cortex. It receives sensory input from all parts of the body and passes them via the thalamic radiations into the sensory cortex. It is connected to various parts of the brain and spinal cord. So we have connection to the spinal cord via the spinothalamic tracts. It is connected to the cerebellum via the cerebello-rubro-thalamic tract. The cerebellar part of this tract passes into the superior cerebellar peduncle. It is connected to the basal ganglia via the thalamus-striate tract, which interconnects the thalamus with the striatum. Gauss structure of the thalamus. The thalamus is rounded in shape with an inferior projection called the pulvina of thalamus. Medial to the pulvina is the methythalamus, which is made up of two important structures as follows: the medial geniculate body and the lateral geniculate body. Superiorly, we have the centromedian nuclear groups of the thalamus, which are interconnected via the third ventricle by the interthalamic adhesion or massa intermedia, that is, left and right parts of the thalamus. It is separated from the caudate nucleus by the striatum terminalis, which contains the terminal vein. It is placed posterior to the caudate nucleus, but medial to the internal capsule, which separates it from the lentiform nucleus. It forms posteriorly the lateral wall of the third ventricle. It has the following functions: sensory input function, which it passes on to the cerebral cortex; emotional function, which is not fully defined; vegetative function, which is equally not well defined. It has the following parts, which in turn carry nuclear groups: dorsal, which is divided into medial and lateral nuclear groups; ventral, which has three distinct nuclear groups; ventral posterior nucleus. This is further divided into ventral posterolateral nucleus, that is VPL for the somesthetic system of the body, and ventral posterior median VPM nucleus for the projections of the trigeminal thalamic tract. Ventrolateral nucleus VL is for superior cerebellar peduncular projection. Then you have ventral anterior nucleus. The lateral part has the following parts: the pulvina, lateral posterior nucleus, lateral dorsal nucleus. Classification, which takes into consideration simple nuclear groups and not the topography, is as follows. There are the following nuclear groups in the thalamus: anterior nuclear group, intralaminar nuclear group, lateral nuclear group, ventral nuclear group, medial nuclear group, midline nuclear group, metathalamus, unclassified thalamic nuclei, and finally thalamic reticular nucleus. Centromedian nucleus, which carries the massa intermedia, is in the intralaminar nuclear group. The thalamus contains the medial and the lateral geniculate bodies. The hypothalamus is a structure placed ventral to the thalamus, and it is separated from it by the hypothalamic sulcus. Aside from its main body, it has the following parts: the mammillary body, tubercinarium, infundibulum. The main part is divided into medial and lateral groups of hypothalamic nuclei, different projections. The hypothalamus receives fibers from the following: cortico-hypothalamic tract. These fibers keep the hypothalamus in check by the cerebral cortex. Thalamo-hypothalamic tract. This tract helps to interconnect the thalamus and the hypothalamus. Retino-hypothalamic tract. This tract interconnects the retina with the hypothalamus, thereby allowing visual activities to influence this highly vegetative functional organ. Hypocampo-hypothalamic tract. This interconnects the hippocampus, which is part of the archicortex, with the hypothalamus. Amygdalo-hypothalamic tract. This tract interconnects the amygdala with the hypothalamus. Its function, just like that of the hypothalamic tract, is mainly reproductive, but also includes taming of aggression and etc. In Clover-Bosse syndrome, described by Clover and Bosse in the 1930s, the monkeys whose amygdala had been removed were found to be hypersexual, docile, and very attentive. Functions of the hypothalamus. This is the main vegetative functional organ of the diencephalon. It is responsible for the control of the autonomic nervous system, the diffuse endocrine system, the endocrine system itself, and also emotional and behavioral mechanisms, since it is a part of a so-called mesolimbic system. Epithalamus. Epithalamus is placed at the back of the diencephalon. It is made up of 4 structures. 1. The pineal body. 2. Habenular trigones or nuclei. 3. Epithelial roof of the third ventricle. And 4. Striae medullaris thalami. Pineal body. This gland is unique because it is both sensory and neural. It contains pseudosensory pinealocytes and endocrine pinealocytes, which have been classified as paraneurons. Endocrine pinealocytes release the hormone which has been identified as melatonin into the third ventricle, a process that is now called ventriculocrania. The hormone functions in circadian rhythm and probably also in reproductive mechanism. Habenular trigones. These contain habenular nuclei which are interconnected via the habenular commissure. The habenular commissure is one of the 2 posteriorly placed commissures of the brain. The other is the posterior commissure, which marks the junction between the midbrain and the diencephalon and is placed below the habenular commissure. The habenular nuclei project fibers into the pineal gland. Also, fibers from the nucleus of the posterior commissure and lateral optic tract and the nucleus of the perirubar area all project into the pineal gland. But the pineal gland itself has no efferent projection anywhere, suggesting it is a bona fide endocrine gland. Striae medullaris thalami. This is made up of several fibers in the diencephalon and the upper part of the midbrain. They are formed by fibers from the various parts of the brain as follows: the globus pallidus, lateral preoptic region, septal nuclei, and anterior thalamic nuclei. Epithelial roof of the third ventricle. This is a bona fide part of the epithalamus. It just simply covers the roof of that ventricle. Subthalamus. This is a small structure in the diencephalon which is connected, strangely enough, not copiously to the diencephalon but to the small brain via the amygdala. It has a subthalamic fasciculus by which it is connected to the thalamus. This fasciculus is connected to the padiludofugal system. The subthalamus is placed ventral to the thalamus but caudal and lateral to the hypothalamus. Welcome to this talk on basal ganglia. Basal ganglia should actually be called the basal nucleus, since it is a collection of nuclear groups which developed at the floor of the lateral ventricle, close to the third ventricle, and around the diencephalon and the lateral wall of the lateral ventricle. They are actually telencephalic structures, having developed from the telencephalic brain vesicle, which in turn comes from the prosencephalon. They have important function in the human body. The basal ganglia is said to be the head ganglion of proprioception in the body. The basal ganglia is, when damaged or diseased, the body loses coordination of movement because it is a strong center for the control of the extrapyramidal system together with the cerebellum. When clinicians refer to the extrapyramidal system, most tend to assume it is the basal ganglia. This is because of a common condition known as Parkinsonism or Parkinson's disease, which is caused by impairment of a part of the basal ganglia. The basal ganglia reaches its highest level of development in birds, where it controls the motor function of this group of animals. When a bird, like the hen, has its cortex removed, it is still able to function as if nothing has happened to it. But once the basal ganglia is damaged, the bird is unable to perform all the intrinsic or instinctual mechanisms such as laying egg, sitting or incubating the laid eggs, courtship, which is a complex, repetitive process in birds, etc. Hence, it is recognized as a center for instinctual mechanisms, whether motor or otherwise. It is therefore not too bad to suggest that the basal ganglia in man may also be responsible for any instinctual mechanism in man, if present at all. The only type of instinct which may be recognized in man is probably the rudiments of temperaments. This shows the differences between temperaments as inheritable traits on one hand, character as modifiable behavior due to environmental influences and personality on one hand, which are conscious behavioral attitudes that usually would not have anything to do with temperament or indeed character. The mesolimbic system described by Papex in 1936, which seems to be related to the human behavior and emotion, has a generous component from the basal ganglia. The basal ganglia consists of two parts, the corpus striatum and amygdaloid nuclear complex. Another division of the basal ganglia recognizes the following parts: archis striatum, which is the amygdala; paleostriatum, which is the globus pallidus; neostriatum, which is the putamen and caudate nucleus. Another name for neostriatum is striatum for short. Another name for paleostriatum is pallidum for short. Pallidum is mainly motor part of the basal ganglia, while striatum is mainly the sensory part of the basal ganglia. Hence, striatum receives many afferent projections but gives out only a few efferent ones. On the other hand, pallidum has many efferent projections but receives only a few afferent fibers. Corpus striatum is therefore made up of the pallidum and the striatum as follows: the caudate nucleus, lentiform nucleus. Lentiform nucleus is further divided into the globus pallidus and the putamen. Striatum. This is the sensory part of the basal ganglia. It receives fibers from the following: nigrostriatal tract. This tract interconnects the midbrain with the basal ganglia, and it manufactures dopamine, which is extensively used in the basal ganglia. The deficiency of this amine would precipitate Parkinsonism. Thalamus-striatal tract. This tract begins from the thalamus and ends in the striatum. Pallido-striatal tract. This tract begins from the pallidum and interconnects with the striatum. A few efferent projections are recognized. 1. Strio-nigral tract. This is between the striatum and the substantia nigra. 2. Strio-pallidal tract. This is important and is a tract that helps to keep information from the sensory part of the basal ganglia to the motor part, that is the pallidum. It forms pencils, and this pencil-like striations on the pallidum is called Wilson's pencil. Pallidum. The afferent or efferent projections are: number 1, striopallidal tract, that is the Wilson's pencil, and number 2, subthalamic pallidal tract or subthalamo-pallidal tract. The pallidum is the motor part of the basal ganglia. It has numerous efferent projections, of which the most important are as follows. Number 1, ansa lenticularis. This project from the pallidum to the forest from where they enter the forest with H2. Number 2, lenticular fasciculus. This project into the forest field H1. Subthalamic fasciculus. This interconnects with the subthalamus. Thalamic fasciculus projects to the thalamus. Pallidotegmental fasciculus. This connects with the pontine reticular formation, which is called the nucleus tegmenti pedunculo-pontinus. Amygdaloid nuclear complex. This complex is divided into the following parts: a dorsal part a ventral part, and other parts. It is also divided into two main nuclear groups, a corticomedial nuclear group and basolateral nuclear group. It is related to the associated nuclei called the periamygdaloid body. The periamygdaloid body is more related to the paleocortex than the basal ganglia. It is a part of a small brain, but its association with the amygdala suggests that, at least in animals where the sense of smell is important, the basal ganglia functions effectively. The cerebrum. The cerebral hemispheres are two, that is, left and right. The two are interconnected via the massive corpus callosum, which is one of the commissural fibers of the cerebrum. But the corpus callosum interconnects the cerebral hemispheres point for point. The word cerebrum can be used interchangeably with cerebral hemisphere, but not with cerebral cortex, for the cortex is a part of the hemisphere. The parts of the cerebrum are as follows. The cerebral cortex, this is the most superficial and also the most sophisticated and rostral of all structures, not only in the cerebrum but also in the whole of the neural axis. Intermediate zone. This zone contains mainly fibers which crisscross and run in various directions. The fibers are either association, commissural, or projection fibers. The intermediate zone may be rightly labeled cerebral medulla. The last is the basal ganglia. Phylogenetic considerations and classification of the cerebrum. We have the following subdivisions based on sophistication of the apparatus. The least sophisticated of the part of the cerebrum is the archicortex or the archipallium. This is followed by the paleopallium or the paleocortex. Then we have the most sophisticated and the newest addition to the cortical apparatus, which is the neocortex or neopallium. The archicortex is the least sophisticated in the cortex. It is the most primitive, appearing first in the lower vertebrates. In man it is made up of the following structures. Number 1, hippocampal formation or hippocampus. Number 2, indusium brisum. Number 3, dentate gyrus. Number 4, fasciolar gyrus. In man it is difficult to say clearly what the function of the archicortex is, but the hippocampal formation is well known to take part in the memory mechanisms, that is, specifically in immediate memory. This is because we know that a person who has a hippocampal disease may suffer from impairment of immediate memory. There are 3 different types of memory mechanisms which are distinct. They are short-term, long-term, and immediate memory. Those who get old find that the impairment of memory is more with immediate memory than the others. Long-term memory is extremely well developed in old age. The archicortex and the so-called juxtalocortex have only 4 cytoarchitectonic layers. The paleocortex. This is the spell brain. It is also called the rhinencephalon. In man, it is not fully developed because man is a microsomatic animal, and his ability to smell is subjugated by his ability to see. He is therefore a social animal, for only what is seen becomes important in social activity and interaction, rather than what is smelled. Olfactory pathway. The pathway for the olfactory nerves begins in the olfactory epithelium of the lateral wall of the nasal cavity. The receptors are attached to peripheral fibers of the bipolar olfactory neurons, which traverse the cribriform plate of the ethmoid. They are about 20 in number. The cell bodies lie in the olfactory bulb. The central process is now synapse, with second-order neurons which enter the olfactory pathway. This pathway is divided into 2, medial and lateral olfactory striae. At the synapse, they form a glomerulus with more than 1 neuron. Medial olfactory striae terminates in the subcallosal gyrus. The lateral olfactory striae carries fibers which terminate in the piriform lobe and the periamygdaloid body. The two structures mentioned, that is subcallosal gyrus and the piriform lobe, are part of what is called the primary olfactory cortex. The third order neuron now projects into the secondary olfactory cortex, called the entorhinal cortex. Fourth or third order neurons issue from the entorhinal cortex to reach the highest centers for olfaction in the hippocampal formation and the insular cortex. Piriform lobe is made up of the following: prepiriform lobe, the prepiriform area, the uncus, and the anterior part of the parahippocampal gyrus. Periamygdaloid body is surrounded or surrounds the amygdaloid nuclear complex. It mediates sensation of smell around the amygdala. Amygdaloid nuclear complex is divided into two: corticomedial nucleus and the basomedial nucleus. It has two main efferent projections. Number one, the striae terminalis. Number two, ventral amygdalofocal projections, which may be also divided into two types: to the hypothalamus through the amygdalo-hypothalamic tract, and then to the thalamus and via the amygdalothalamic tract. Because of the intimate connection of amygdala with these diencephalic structures, its function is similar in the control of visual activity. For example, the stimulation of amygdala leads to ovulation. It is also said to control gonadotropin levels together with the hippocampus. Smell types. There are 7 primary smells recognized. They are as follows: number 1, camphoraceous; number 2, ethereal; number 3, musky; number 4, floral; number 5, putrid; number 6, pungent; and number 7, pepperminty. The neocortex. This is the most sophisticated of all the parts of the brain and the most sophisticated of its telencephalic structures. It is massive in man, and the process by which it becomes this massive is usually termed prosencephalization, which reaches its peak in man. For its function, it is provided with gyri, which increase the surface area of a superficial part of a complex, or of a cortex. It is divided into lobes as follows: frontal lobe, parietal lobe, temporal lobe, and occipital lobe. There is also buried lobe called the insular cortex. Also, there is a functional lobe involved in the process of emotions and behavior. This is called the limbic lobe. The frontal lobe. The frontal lobe of the cortex seems to be the one that is most highly developed for cognitive mental functions, which may include thinking, rationality, memory, abstract and concrete thoughts. It is also thought to have a function in emotions and personality formation. This is because in 1936 a surgical procedure was designed to cure permanently insanity. This procedure was termed prefrontal leucotomy, which literally means cutting through a white matter of the frontal lobe, thereby separating the frontal lobe from its prefrontal connections, because it is believed that the prefrontal part of the lobe is responsible for personality and behavior. Unfortunately, the preparation went out of hand, so that the individual loses his personality and becomes docile. He behaves like a vegetable and is unable to reason out things himself. He performs anything he wishes without worrying about its consequences. By the 1950s, the procedure was stopped because of its barbaric nature, and also because of the discovery of new psychotropic drugs which are able to make a psychotic man perform his functions satisfactorily. The above abandoned procedure is called psychosurgery. The prefrontal area is therefore responsible for psychic motor function. That is to say, it helps the individual to his individualistic reactions to his environment. The way he will behave is peculiar to him and not to anyone else. This individuality is one of the attributes of the human race. The area that takes part in the emotional and pre-evolutional mechanisms in the prefrontal region are the areas 6 to 8. Area 4 is part of the frontal lobe and it takes care of voluntary motor function. In this area, the entire human body is structured around the precentral gyrus, which is the motor area. This gyrus is placed between the precentral sulcus and the central sulcus. The gyrus has a representation which is called the homunculus. This homunculus has a large representation of the hand at its inferior aspect to demonstrate the importance of the hand generally in motor function. The buttock is placed at the edge of the superior aspect of the precentral gyrus so that it extends from the lateral to the medial aspect of the frontal lobe. The rest of the frontal lobe is divided into superior, middle, and inferior frontal gyri. There is also gyrus rectus, which is placed at the basilar part of the brain overlying the olfactory tracts. Welcome to the next and the concluding section of the cerebrum. We shall start this section by talking about the parietal lobe. This lobe is recognized for sensory activity. It has the post-central gyrus, which carries the areas 3, 1, and 2. The sensory fibers are projected into it from the thalamic radiations. The rest of the parietal lobe is divided into superior and inferior parietal lobules. The inferior lobule is further divided into supramarginal and angular gyrus. The areas outside the sensory cortex are the psychic areas which control the way and manner that we perceive sensory information from our environment. The occipital lobe. This is responsible for visual activity. It has the visual cortex, which is in the areas 17 to 19, although most of the areas include also psychic components to what we see. The occipital lobe is separated from the parietal lobe by an internal parieto-occipital sulcus. This sulcus forms a Y-shaped sulcus with the carcinoid sulcus, whose bed contains the visual cortex. In the occipital lobe, we also have the cuneus and the lingual gyrus. Temporal lobe. This lobe is mainly known for auditory, vestibular, and gustatory functions. Areas 41 and 42 are considered auditory, while area 43 of the temporal lobe is gustatory. It has the following gyri: superior temporal gyrus, middle temporal gyrus, and inferior temporal gyrus. There are certain parts of the brain which extend their gyri beyond the ordinary boundaries of the cortical lobes. We have a lateral and medial temporal occipital gyri in the basis of the brain. The parahippocampal gyrus, subcallosal gyrus are also found. The insular lobe. This insular lobe is considered to be part of the limbic system and is therefore a part of the emotional apparatus of the brain. It is buried in the cerebrum lateral to the basal ganglia. Its white matter is the extreme capsule, which is directly medial to it. Medial to the extreme capsule is the claustrum, followed by the external capsule and then the lentiform nucleus containing globus pallidus and the putamen, which is also part of the basal ganglia. Intermediate zone of the cerebrum. This contains fibers as follows: number 1, associating fibers; number 2, projection fibers; number 3, commissural fibers. Association fibers are divided into two: long and short association fibers. There are a number of examples such as uncinate fasciculus, arcuate fasciculus, and the superior longitudinal fasciculus. Projection fibers. Examples are the corona radiata, which later becomes the internal capsule. Commissural fibers. These interconnect the sides of the brain. You have the anterior commissure, which is a telencephalic structure, and also the corpus callosum. The corpus callosum can be divided into the following parts: genium, splenium, and rostrum of the corpus callosum. It is the largest commissural fiber in the brain. thalamic radiation. These are projection fibers which accompany the internal capsule across the diencephalon and the basal ganglia. They arise from the thalamus and project upwards to reach the sensory cortex. They are divided into the following parts: anterior thalamic radiation, which accompanies the anterior limb of the internal capsule; posterior thalamic radiation, which accompanies the posterior limb of the internal capsule together with the retrolenticular part of the internal capsule. Also, occipitopontine fibers are associated with it. Superior thalamic radiation, which is the last, projects from the thalamus to the parietal lobe. This is the very last one, is the inferior thalamic radiation, which projects fibers from the thalamus to the temporal lobe. It is accompanied by the temporopontine fibers. Brodmann's area of the cortex. These include the visual cortex from 17 to 19, gastratic cortex areas 43, sensory cortex areas 3, 1 and 2, motor cortex area 4, premotor cortex areas 6 to 8, and auditory cortex areas 41 and 42. All these areas tend to have 6 cytoarchitectonic layers in their cortex as follows. The most external is the molecular layer, followed by the external granular layer, followed by the external pyramidal layer, followed by the internal granular layer, followed by the internal pyramidal layer, and finally you have the fusiform layer. The names of these layers are the same names given to the cells found in the larynx. Thus we have granular cells found in the granular layers, fusiform cells found in the fusiform layer, and pyramidal cells are found in the pyramidal layer. Functions of the cerebral cortex. The cortex has 2 main functions: recognition and performance. Recognition is a sensory phenomenon, while performance is a motor phenomenon. Inability to recognize is called agnosia. Inability to perform is called apraxia. There are several examples of apraxia and agnosia in life. We have the expressive aphasia, which is a form of apraxia, and also receptive aphasia, which is a form of agnosia. The ventricles of the brain have the following divisions: 2 lateral ventricles, a 3rd ventricle, and a 4th ventricle, which communicates with the central canal of the spinal medulla. Lateral ventricles. The 2 lateral ventricles have the following divisions. Number 1, anterior horn, which projects into the frontal lobe. Number 2, a posterior horn, which projects into the occipital lobe. Number 3, inferior horn, which projects into the temporal lobe. The anterior horn has a roof made up of the corpus callosum, and the lateral wall is made up of the caudate nucleus. It projects into the body of a lateral ventricle at the trigone of a lateral ventricle. The trigone of a lateral ventricle communicates with the body, the occipital horn, and the temporal horn of the lateral ventricle. The temporal horn has a medial wall made up of the hippocampus. Its dorsolateral extreme is formed by the amygdaloid nuclear complex. The occipital horn has a roof made up of the tapetum. The lateral ventricles communicate with the 3rd ventricle by the interventricular foramina. The 3rd ventricle has a lateral wall made up of the thalamus. It communicates with the midbrain, which surrounds the cerebral aqueduct. This aqueduct links with the 4th ventricle. At the rostral extreme of the medulla, the 4th ventricle opens into the 2 lateral apertures called the foramina of Luschka. Meninges. The meninges are the coverings of the structures of the central nervous system, that is, the spinal cord and the brain. The meninges are usually divided into cranial and spinal meninges, depending on whether they cover the brain or the spinal cord. they can be further divided into three parts, three types, or three coverings. The most external is the dura meter. This is followed by the arachnoid meter and then the pia meter. The dura is also called the Purkinje meninges. It is the thickest of all the coverings of the neuraxis. It is divided into two portions, the more superficial part of which is attached to the cranial periosteum. This is the cranial dura. The cranial dura is followed by a deeper dura directly covering the brain substance. The space that separates the dura and the arachnoid meter is the subarachnoid space. The subdural space. This space is important clinically because of the possibility of hemorrhage as a result of a tear in the branches of the arteries within the space. This type of hemorrhage is known as subdural hemorrhage. Hemorrhage may also occur clinically into the space above the dura by a tear in the branches of the meningeal arteries that supply the dura itself. It is then called epidural hemorrhage. The arachnoid and the pia are thin and are applied to the surface of the brain very closely. They are together called the pia-arachnoid complex or the leptomeninges. This leptomeninges is very thin, but there is also a potential space between the two meters called the subarachnoid space. Cerebrospinal fluid, CSF, is contained within the subarachnoid space. This space also contains subarachnoid granulations which allow for the flow of CSF in a unidirectional fashion. The CSF flows through the arachnoid granulations into the main bloodstream by entering intravenous sinuses around the brain. The subarachnoid space communicates with the ventricular system of the brain through two lateral foramina of Luschka and one median foramen of Magendie. CSF therefore moves freely within the ventricular system, communicating with the subarachnoid space. Hemorrhage may occur in the subarachnoid space, and this is then called subarachnoid hemorrhage. Cisterns are spaces containing large amounts of CSF placed between the pia-arachnoid complex and the dura. The largest cistern in the brain is the cisterna magna, placed between the cerebellum and the medulla oblongata. This is the cerebellomedullary cistern. Pontine cistern is placed anteriorly between the two halves of the pons. The superior cistern is placed around the posterolateral part of the midbrain. Other cisterns are the interpeduncular cistern around the cerebral peduncle, chiasmatic cistern around the optic chiasm, and the lumbar cistern in the spinal cord. These cisterns are important because they can be used in outlining the brain in radiological imaging and in pneumoencephalography. The limbic system was described by Broca for the first time in 1878 as grand lobe limbic. It is a system that helps to control human behavior and emotions. It is made up of a central core of structure which is called the limbic lobe. This lobe consists of the following: subcallosal gyrus, cingulate gyrus, para-hippocampal gyrus, and hippocampal formation. Cortical contribution to the limbic system as follows. Aside from the limbic lobe, the following structures are associated with the limbic system: temporal cortex, prefrontal lobe, insular lobe, and orbital gyrus. But the limbic lobe is also supported by other structures in the brain, forming what is called the mesolimbic system because of the involvement of the mesencephalon. It is sometimes called the septo-hypothalamo-mesencephalic continuum. Other parts of the cortex which are in the limbic system are sometimes called the juxtalo cortex. The basal ganglia structures are also involved in emotion and are therefore part of the limbic system. The encephalic structures include the thalamus, hypothalamus, and subthalamus. The thalamus provides pain and possibly pleasure centers. The hypothalamus is the center for endocrine emotional activation, for it seems to have a direct continuity with the visceral endocrine periphery. The midbrain is believed to take part in the limbic system through its reticular formation, especially the extension of the nucleus tegmenti pedunculopunctinus, the visceral endocrine periphery. Emotional mechanisms must be accompanied by activation of the visceral endocrine periphery, such as lacrimation, laughter, crying, wailing, moaning, gnashing, and all other factors associated with emotions. They must be activated by certain centers. These appear to be autonomous and therefore may have something to do with the autonomic nervous system. Hence, the mesolimbic system seems to have a receptor mechanism and an effector mechanism. It has been postulated that there is very little connection from the cortex to the limbic system, so that the limbic system has very little control from the cortex. Even though this control is little, the cortex still maintains some amount of control over the instinctual mechanisms of the limbic system. So, the human brain, the cortex, learns all life. The human brain learns to do what it is right or what he believes is right to do, rather than what he thinks he wants to do. But in mental illness, it is postulated that the little connection between the cortex and the limbic system is completely cut off, so that the limbic system now functions as if it were on its own. When information comes from the cortex, which is to be directed to the limbic system, the connection does not take place, and that information does not stimulate any of the centers, whether pleasure or pain centers in the limbic system. Hence, the mentally ill can have spontaneous discharge of the limbic system structures, either pain or pleasure. In the case of pain, depressive illness occurs. In the case of pleasure, mania or hypomania may be the consequence. The optic tract. The visual fields can be divided into temporal portion and nasal portion. The temporal portion of the visual field projects on the nasal portion of the retina. Information then proceeds from this nasal portion through the optic nerve to the optic tract. Information from the nasal visual field projects onto the temporal retina. At the optic chiasm, fibers carrying impulses from the two fields of the retina document partially. Lateral fibers proceed along the same line while medial fibers cross to the other side. Hence, the optic tract consists of contralateral fibers from the nasal retina carrying impulses from the temporal field, and each lateral temporal fibers carrying information from the nasal field. The optic tract then moves around the brainstem to terminate in the lateral geniculate body. Second-order neurons move from the lateral geniculate body as optic radiations within the sublenticular part of the internal capsule. Some fibers proceed to the superior colliculus to form the brachium of the superior colliculus. The superior colliculus observes primitive visual reflexes. The fibers of the optic radiation then terminate in the visual cortex to form the geniculo-calcarine tract that calls along the bank of the calcarine sulcus to terminate in the area 17 of the medial occipital cortex. This is the visual cortex. Clinically, blockage can occur in any part of the tract. A complete transection of the tract before the optic chiasm causes total blindness of the eye of that side. A lateral section at the optic chiasm will lead to an ipsilateral nasal hemianopsia. A section posterior to the optic chiasm at the optic tract causes a left-sided homonymous hemianopsia if the section is on the right side. A section across the optic chiasm in the sagittal plane leads to bitemporal heteronymous hemianopsia. This usually occurs in pituitary tumors because of the close relationship between the hypophysis cerebri and the optic chiasm. Blood supply to the brain and the spinal cord. The spinal cord is supplied by two arteries, anterior spinal artery and posterior spinal artery. They provide radicular arteries which enter the spinal cord substance. The brain is supplied by two main arteries also, the internal carotid artery and the vertebral artery. These two arteries form an arterial circle of Willis at the ventral aspect of the brain. The internal carotid artery consists of the following parts: the cervical part, interpetrosal portion found in the petrosal sinus, intracavernous portion found in the cavernous sinus, the supraclinoid portion found above the anterior clinoid process. Most of its branches to the brain arise from the supraclinoid. The vertebral artery from the neck region transverses the foramina transversaria, except for the first cervical vertebra of the four upper 6 cervical vertebrae, in order to approach the basis pontis, where the two vertebral arteries join to form the basilar artery. The basilar artery communicates rostrally with the branches of the internal carotid artery to form the arterial circle of Willis, which is the main supply of the blood to the brain. The arterial circle has two terminal branches of the internal carotid artery as part of it, and they are the anterior cerebral artery and the continuation of the internal carotid artery itself, which is the middle cerebral artery. The two anterior cerebral arteries from right and left communicate in front by an anterior communicating artery, which completes the circle by joining the posterior cerebral branch of the arterial circle of Willis. The anterior and middle cerebral arteries supply the following structures by their branches: number 1, frontal lobe; number 2, upper part of the temporal lobe; number 3, parietal lobe; number 4, anterior portion of the basal ganglia; number 5, anterior portion of the thalamus. The posterior cerebral artery supplies: number 1, posterior part of the basal ganglia; 2, posterior portion of the thalamus; 3, occipital lobe; 4, medio-inferior portion of the temporal lobe. The arterial circle of Willis is formed at the base of the brain to support the blood supply to the brain, and the following are part of it: number 1, anterior communicating artery, which interconnects the anterior cerebral veins, anterior cerebral arteries; posterior communicating artery, proximal part of anterior cerebral artery, proximal part of middle cerebral artery, and the proximal part of the posterior cerebral artery. The arterial circle of Willis has branches which are circular, called circular branches, and they are divided into 4 groups: anterolateral group, anteromedial group, posterolateral group, and posteromedial group. The most important clinically is the anterolateral group, which forms the lateral striate artery called the artery of cerebral hemorrhage, because it supplies most of the internal capsule and is most easily damaged by the hypertensive stroke, causing pressure on the internal capsule and loss of blood supply to the posterior limb of the internal capsule, thereby damaging the corticospinal tract. The contralateral side of a lesion will be affected and paralyzed. This artery of cerebral hemorrhage also supplies the region surrounding the posterior limb of the internal capsule, such as the thalamus, and puts pressure on the same as a result of hemorrhage. Some of the branches that supply the thalamus may also supply the internal capsule and hence may cause paralysis if damaged. They are thalamo-geniculate artery, which supplies the thalamus and the medial and lateral geniculate bodies. Anterior choroidal artery, which supplies the posterior edge of the internal capsule and may cause problems with affection of the internal capsule itself. The internal carotid artery begins at the level of the upper border of the thyroid cartilage. It is separated from the external carotid artery by the following structures: pharyngeal branch of vagus, the styloid process, stylopharyngeus, styloglossus, and glossopharyngeal nerve. It moves upwards from this level and is separated from the internal jugular vein by the vagus nerve. It proceeds upwards to reach the base of the skull. Snubbing the foramen lacerum, it enters the carotid canal. It emerges at the middle cranial fossa, where it projects upwards above the clinoid process to form a siphon that projects at right angles to its proximal end. It finally divides into its two terminal branches, anterior cerebral artery and middle cerebral artery. It is divided into four parts along its course. The supraclinoid part gives off the following branches: anterior communicating artery, which interconnects the anterior cerebral arteries; anterior choroidal artery, which supplies the posterior edge of the internal capsule; posterior communicating artery; anterior cerebral artery; and then middle cerebral artery. These last two are its terminal branches. The anterior cerebral artery supplies the inner aspect of the cerebral hemispheres. Its branches include medial stride artery, orbital branch for the orbital plate, callosal-marginal artery, which supplies the corpus callosum, and then pericallosal artery. The middle cerebral artery, on the other hand, supplies mainly the external part of the brain and has the following branches: orbitofrontal artery, anterior parietal artery, posterior parietal artery, posterotemporal artery, anterotemporal artery, Rolandic artery, and the pre-Rolandic artery. And the Rolandic artery supplies the central sulcus, and the pre-Rolandic supplies the precentral gyrus and sulcus.